our environment is constantly changing. The sun rises and falls in a day, and the seasons change over a year. In addition to this natural cycle, increases in carbon dioxide and other greenhouse gases caused by human activity in the burning of fossil fuels is increasing global temperatures, bringing more extreme and unpredictable weather patterns. These changes to our environment affect all life on this planet. In the Plant Molecular and Cellular Biology Laboratory, led by Dr. Joanne Chory at the Salk Institute, we seek to understand how plants sense changes in their environment, how they respond to these changes, and ultimately, if plants can be part of the solution to our climate crisis. To study plant responses to the environment, we take advantage of different plant species. For example, the mustard weed, Arabidopsis thaliana, is small, easy to grow, has a short life cycle, and a small, compact genome, which is easy to mutate and genetically modify. This species is the workhorse of the plant biology community. Basic principles which we learn in this plant can then be translated to other crop species on which we work, such as tomato, oilseed and vegetable varieties of brassica, and rice. Other more unusual species, such as the carnivorous plant Venus flytrap, are used to learn about how plants evolve mechanisms to sense specific environmental stimuli, such as in this case, the touch of insect prey, and how these touch signals are converted into an electrical signal that propagates across the leaf to induce movement and prey capture. This electrical signaling is a feature shared with other plants in response to herbivore-induced wounding. For much of our work, we need to carefully modify a plant's environment and track its growth in response. In our climate control facility, we can modify the wavelengths and intensity of light, the humidity, and the temperature at which a plant grows to simulate real-world conditions. Further control of the environment in which we grow our plants can be achieved by growing seedlings in tissue culture conditions, which also facilitates easy measurements of plant root and shoot architecture.
One of the goals in our lab is to understand the molecular basis by which a plant senses and responds to these environmental conditions. What are the genes involved? To address this, we screen through mutant libraries for plants that have altered growth patterns or conduct experiments in which we track changes in gene expression as a result of an environmental stimulus. When we have identified the DNA sequence of the gene of interest, we can further manipulate the plant genome to test our hypotheses. Like other plant molecular biology labs, we take advantage of specialized bacteria that naturally live in the soil, with which we can introduce foreign DNA into the plant genome. Using this system and gene modification tools, we can introduce specific lesions into the DNA or express modified proteins that can be visualized on a microscope. Using fluorescence microscopy, we can image in which tissues a particular gene is expressed, and where within the cell the protein might localize and function. This can give us clues as to the biological processes within which a gene and its protein product are involved. Lastly, plants with modified genomes are returned to different environments, and we carefully track how they respond. Key manipulations may be replicated in crop varieties to further test if we can make predictable changes to crop plant growth in specific environments. Ultimately, our methods determine the basic genetic underpinnings of how a plant grows in diverse environmental conditions. The next big challenge being addressed by the Salk Institute is to see if we can now use plants to alter our environment on a global scale and reduce the levels of the greenhouse gas carbon dioxide in the atmosphere.
Now that you've seen what we do at Salk, I'm going to show you a little bit about our off-campus greenhouse. Follow me! Welcome to our greenhouse. To accomplish this goal of combating climate change through plants, we need to increase their carbon sequestration ability, which is their ability to suck up carbon from the atmosphere. Here you can see we grow a variety of plants. To do this, we have a state-of-the-art 12,000 square foot greenhouse where we grow our crop plants and study them for indication that they'll be able to sequester more carbon. In order to make sure our watering is controlled, we have an automated irrigation system. The water flows through the pipes here and through the emitters out into the spitters and into the plant pot. We are constantly monitoring the amount of nutrients or water that a plant leaches out. Now let me show you our lab. Here we have a freeze dryer, a plant drying oven, a growth chamber, a biosafety hood, water bath station, an autoclave, and our X-ray machine. Continuing on, we have our scanning stations, a workstation, our refrigerator, and our imaging cart. One of the most important things we do in the greenhouse is process the plant samples that we have grown. Phenotyping the plants is one of the most important aspects of the project, as it allows us to identify what we want to move forward with. When the plants are ready to be processed, we will excavate the entire plant from its pot and wash the root system. The first step in this is to dump the pot in a vat of water and rinse the roots of the substrate they've been grown in. Once the roots have been thoroughly cleaned, we will then dry them out and sieve the water that we use to wash the roots in order to capture any remaining roots that may have fallen off in the process. Once the plants have completely dried out, we will then weigh the samples and collect data on the roots and the shoots. Depending on the crop, we'll dry the plants in a multitude of different ways. For most crops, we air dry, hang dry, and then put them in our drying oven for a certain duration depending on the crop type. Depending on our needs, the plants have one of two fates. They will either get processed for data, or we will seed bulk them so that we can retain seed for future studies. When seed bulking Arabidopsis thaliana, we will bind and stake the plants once they have bolted and are creating flowers. When they've reached the appropriate growth stage, we will allow them to dry out completely, and once they are dried, we will harvest them. Once the Arabidopsis has sufficiently dried, we will cut the base of the plant and remove the stake and the bag and the plant altogether. Then we'll take the plant out of the bag and we'll put it in a sieve. The plant is then crushed in the sieve and we separate the excess dried plant material and the seeds at the same time. One problem we run into with studying the roots is that you generally have to excavate and kill the plant to get data on the root system. In order to study the root system without killing the plant, we have an incredible piece of equipment that x-rays the root system and generates an image of inside the pot without having to excavate the plant. The machine slowly turns the plant and takes approximately 3,000 images while the plant is turning. It's technology like this that propels us forward in our research and allows projects like the Harnessing Plants Initiative to be possible. In order for a project like this to be successful, we have to work with a diverse set of individuals in order to solve our global issue. After all, global issues require global, scalable solutions. Ultimately, at the Salk, we are working hard to address how plants sense and respond to their environments, and if in turn we can take advantage of the power of plants to tackle climate change for a better planet.